In this tutorial, we'll look at another pathogen group, that of the bacteria. We'll start on this page here of the tutorial. It shows a couple of photographs taken through microscopes. This one here is taken under a light microscope, probably at around about 400 to 1,000 times magnification, I would say, under a light microscope. This photo here is taken under an electron microscope, and you can see the bacteria, which in this case are bacilli bacteria, lined up here, probably in the process of reproduction since they're still joined end to end. So this is possibly under several 100,000 times magnification. So these are the same objects, or very similar to these bacteria here. Okay, let's have a look at the basic structure of a bacterium. Alright, bacteria belong to the group that we call the prokaryotes. So they're prokaryotic cells, or they belong to the group prokaryotes. So bacteria are the only members of this particular classification. All other cells belong to the group that we call the eukaryotes. So they're prokaryotic cells, and what defines prokaryotic cells, as you probably already know, is that they're very small, very simple in structure. They have no nucleus, so their DNA is a single circular strand that sits in the cytoplasm, and there are no membrane-bound organelles. So there are no mitochondria, there's no chloroplasts, there's no vacuoles, no Golgi apparatus, no endoplasmic reticulum. So any organelle that has a membrane will not be found in bacteria. Nevertheless, they are cells and they carry out all the same functions as most cells. They carry out respiration, so they'll carry that out in the cytoplasm. They carry out protein synthesis, so they have ribosomes, but remember ribosomes don't have membranes. And they have cytoplasm. They have a cell wall, although the cell wall in a bacterium is made out of slightly different material to the cellulose of plant cells. So on the left here we can see a diagram of a generalized bacteria, a rod-shaped bacteria. Many bacteria have a whip-like structure called a flagellum, or the plural flagella, that assists their movement through a liquid medium. Bacteria generally have to live, not generally, they have to live in a liquid medium, otherwise they'll dry out, dehydrate. So you can see the cell wall labelled here, the cytoplasm inside the cell here, various ribosomes to help with protein synthesis, little structures called the pili, which also assist movement, and so on. So a fairly basic, simple structure. In terms of their general features, they are generally defined by having three basic shapes, referred to as the cocci, bacilli and spirilla. We'll have a look at those in a little more detail in just a moment. Unlike viruses, which are all pathogenic, so all viruses cause disease because they destroy their host cells, not all bacteria are pathogenic. There are some bacteria that do not cause disease and quite happily live on the surface and in the inside of many organisms, including yourself. There would be millions of bacteria that live in your gut that assist with the production of vitamins and other essential components that you need in your body. So they live in a symbiotic relationship with you. There are millions of bacteria that live on the surface of your skin, particularly under your arms and in other areas where the warmth and the moisture assist their growth. And it's the bacteria that often cause the gases that we associate with bad odour. Generally, those pathogenic bacteria, the ones that cause disease, do so because they in some way disrupt the host cell of the organism that they've infected, often through the production of very powerful toxins or poisons. And it's these toxins that often in some way disrupt the functioning of the host system. Fundamentally, bacteria are often identified initially through a very famous stain called the Gram stain, not often, always with a capital G, named after Mr. Gram. And the Gram stain differentiates all bacteria on the planet into two basic groups. Those that are described as gram-positive, which always stain a purpley blue colour, and those that are then defined as gram-negative, which always stain a pinky red colour. And so scientists who work with diagnosing bacterial infections always start with a gram stain to make this basic differentiation. Let's have a look at the basic shapes of bacteria. As I said in the previous section, that bacteria generally come in three different shapes. The colours that you see here in these photographs, which look like they've been taken under an electron microscope, are not natural to the bacteria. Bacteria will be transparent like all cells. They won't have a colour to them unless they're photosynthetic, in which case they'll have some chlorophyll inside them. But generally the cells are clear, so what you're looking at are stains that have been put into the cultures so that they become visible and clear under an electron microscope. So don't be fooled by the colours. So the three basic shapes of bacteria is spherical, and we refer to these as Acoccus bacteria, that is the singular, or the plural is cocci. So they have basically a spherical shape. The second group are the ones that are described as a bacillus, which is singular, or rod-shaped, or the plural is bacilli. And the third shape is the spiral-shaped bacteria, Spirillus, which is singular, or Spirilli, plural. And you can see the spiral-shaped here, the rod-shaped here. So all the bacteria on the planet more or less fall into these three categories of shape. Some of the main diseases caused by bacterial infection are listed here. Some of them I'm sure you'll recognise because they're commonly known. Of course, tetanus, which the majority of people are already vaccinated against because it's a deadly bacterium. It produces one of the most powerful toxins or poisons, which is a nerve toxin known to human beings. In the italics here is the scientific name of the bacteria that causes tetanus, which is the disease. So Clostridium tetani. A very, very close cousin of the tetanus bacterium, and you can tell this since it has the same genus name, is Clostridium botulinum, which causes botulism, this bacterium produces perhaps one of the most powerful toxins known to human beings, next to tetanus. Botulism, of course, is the source of Botox. So humans have harvested the botulism toxin and in very, very, very tiny quantities now injected into themselves. It's a nerve toxin, causes paralysis, and essentially the effect of Botox is to paralyse small sections of the muscle and hence remove wrinkles in the skin. Ultimately the body will break the Botox or the toxin down, and so it needs to be injected continuously. Pneumonia, bacterial pneumonia, is very, very dangerous, caused by bacterium pneumococcus, and can be a deadly bacterial disease. Diphtheria can be deadly. A very famous one, tuberculosis, is a very, very difficult bacterium to get rid of. Forms very tough spores and can be very difficult to remove from the lungs, caused by Mycobacterium tuberculosis. Food poisoning is very common, caused by multiple different kinds of bacteria. Commonly though, you may read about outbreaks of Salmonella. Salmonella typhi causes food poisoning which can lead to death in extreme cases. Staphylococcus aureus, or golden staph, is another source of food poisoning amongst other infections. Many throat infections are caused by bacteria called Streptococcus. Typhoid fever, syphilis, chlamydia, which are sexually transmitted diseases, the last two, all caused by bacteria. And of course there are very famous ones like Yersinia pestis, which of course causes the bubonic plague, which killed roughly a third of Europe's population in the 15 and 1600s and earlier. Bacteria reproduce very simply by a process referred to as binary fission. Simply means that the bacterium divides in two to create two new cells. So the chromosome gets duplicated the cytoplasm divides in half, and from one cell you get two identical cells. So it's a form of asexual reproduction. Under ideal conditions, binary fission can occur once every 20 minutes. So you can start from one bacterium and end up with millions in only a matter of hours through exponential reproduction. So you might see growth rates look something like this. Treatment of bacteria is through a very famous set of chemicals that were discovered by accident many years ago in the early part of the 20th century. And of course these chemicals we refer to as antibiotics. The very first antibiotic that was discovered by accident was the antibiotic penicillin. Very, very famous. A miracle cure. Penicillin is a chemical that's capable of disrupting the bacterial cell membranes and hence killing a wide range of bacteria. Over many years, many bacteria have become resistant to penicillin, and so derivatives or slight changes were made to the molecule, and other antibiotics were discovered in nature, and then synthetic antibiotics were also produced, ones like tetracycline, cyclosporin, and there are many others, methicillin. Today there are many different types of antibiotics, some more powerful than others. But as bacteria develop more and more resistance and tougher resistance to antibiotics, doctors are finding they have to use stronger and stronger antibiotics. And there are now some bacteria that can be resistant to all the major antibiotics that we throw at them and are very, very difficult to kill. Here's a couple of agar plates showing the source of the antibiotic penicillin. Penicillin is actually the chemical or toxin made by a fungus called Penicillium. So this is the fungus Penicillium, which is actually a mould. You can see the mould growing here on the agar plate. The mould creates or produces a chemical, which is an antibacterial chemical, which is secreted out into its environment. This is a bacterial streak here. So there's a bacteria growing on an agar plate, and you can see around the fungus what's called the zone of inhibition, where the chemical has killed any bacterial cells, so they're incapable of reproduction. Out beyond the zone of inhibition you can see bacterial colonies that have grown up on the agar plate. This was discovered by accident many years ago. The chemical, once it was purified, became, of course, the very famous antibiotic penicillin, and was injected into human beings or taken orally, and was discovered to be able to kill many, many bacterial infections.